Now I request the another speaker, last speaker for today on Thalassemia Day, Dr. Vandana Arya, who is the consultant and associate professor, Molecular Medicine Lab, Department of Hematology, Sir Gangaram Hospital, New Delhi. She is in charge of Molecular Medicine Lab in Sir Gangaram Hospital, which provides state-of-art diagnostic services and cutting-edge research. She has done research and training in Department of Medical Genetics, Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow, and her expertise are from molecular pathology of hematological and solid cancer, benign hematology, thalassemia, and hemoglobinopathies. She has attended various conferences and she has got more than 50 publications into her credit. She has got supervised as a PI 9 research project, 8 intramural grants, and 1 extramural grant. So I request Dr. Vandana Arya to go and present the talk on the topic Unraveling Thalassemia, Genetic Determinants, and Diagnostic Advancement. I welcome Dr. Vandana Arya.
Thank you, Dr. Harsh, for a kind introduction. Good evening, everyone. I'm going to cover the molecular aspects of the thalassemia. So the topic I have chosen is Unraveling Thalassemia, Genetic Determinants, and Molecular Diagnostic Advances. So first, I would like to tell a little bit about basics of thalassemia. So as everyone is aware about, thalassemia is the commonest monogenic disorders. It's a single gene disorders, and worldwide, approximately 7% of the population carries thalassemia gene. It's a recent published data which shows this kind of incidence. And thalassemia is a group of disorder which is the blood disorder, inherited disorder, and affecting hemoglobin production. It is characterized by the quantitative defect in the globin gene. So the prevalence of thalassemia in Indian subcontinents is approximately 3 to 4%, which is accounting for approximately 35 to 45 million career in our country. So this particular world map showing the thalassemia hotspot areas all over the world. The main areas are Mediterranean region, Southeast Asian regions, Middle East, Africa, and China. This whole darker portion known as a thalassemia belt. So inheritance patterns in thalassemia it's an autosomal recessive manner. Inheritance means 2 defective chromosomes are needed to cause the symptomatic disease. Means both the parent should carry mutation in a carrier form. The carrier frequency definitely varies between geographical distribution of the population, as well as the ethnicity of the population. The risk assessment, as Dr. Sabina and Dr. Swati has already told, that the child, if both the parents are affected with the thalassemia mutation, there is 25% chance to have a affected baby. And there is a genetic counseling required for the people who are at risk for the carrier, for the having the child. So now you can see that the thalassemia, it's a hemoglobin structure. Everybody is aware about hemoglobin is made up of a 2 component, heme plus globin. And heme is the iron, globin is the part which our body synthesize. You can't take that from the outside. So our body is going to synthesize this hemoglobin, and overall our lifespan, different kind of hemoglobins are being produced. In embryonic phase, there is a different kind of hemoglobin. Then there is a fetal hemoglobin, and then there is adult hemoglobin. This structure, you can see there is a tetramer, there is a 2 alpha chain and 2 non-alpha chain. These non-alpha chain in fetal hemoglobin is known as a gamma chain. Then this tetramer is known as the alpha and gamma. And when the adult hemoglobin is being produced, adult hemoglobin is HbA and HbA2. HbA will be made up of 2 alpha chain and 2 beta chain, whereas HbA2 will be made up of 2 alpha chain and 2 delta chain. So the whole sort of thalassemia pathophysiology is based on the ratio of alpha and non-alpha chain. This should be 1:1. In case because of the any defect in globin chain synthesis, this ratio would get disrupted. In that scenario, it will cause the disease. So thalassemia types is two type of thalassemias are there: alpha thalassemia, beta thalassemia. They are based on the defects in respective globin chain. In case the defect is present in the alpha gene, this is known as alpha thalassemia. In case defect is there in the beta chain, that is known as a beta thalassemia. Usually alpha thalassemia is caused by the big deletions, mutations in the 2 alpha gene, which is known as HBA1 and HBA2. Whereas in beta thalassemia, mutations occurs basically in the HBB, which is the scientific name of beta hemoglobin gene, and multiple mutations are there in the hemoglobin, this gene. The severity in the alpha thalassemia ranges from the silent carrier to the a major anemic patient, whereas in beta thalassemia, patient will be either transfusion independent to the severe form of transfusion dependent thalassemia major. So this diagram shows the beta thalassemia pathophysiology. You can see there is in case patient is not having, individual is not having any kind of mutation, the beta globin gene will be intact. There will be nothing like any reduction in the globin chain. But in case any mutation is there in the beta globin chain, then there will be reduced production or absolutely no production of the beta globin chain. That's how alpha beta gene ratio will disrupted and it will get increased, and because of that excess alpha chain will get precipitated. These free alpha chains definitely get precipitated on the RBCs, and ineffective erythropoiesis takes place, and finally apoptosis of the RBCs and hemolysis. The phenotype of beta thalassemia is 3 type of main beta thalassemia traits are there. Thalassemia trait, which is also known as the thalassemia minor. This is the asymptomatic condition where only one chromosome will be affected. It can present at any age. Generally, there is no need of blood transfusion. Thalassemia major is the most severe form of thalassemia where the patient will be presenting with the symptoms in the 6 months to 1 year of age, and regular transfusion is required. Thalassemia intermedia is basically a clinical entity which is somewhere lying between thalassemia trait and thalassemia major. It can be based on the genotype. It can be asymptomatic to severe form of anemia. And the presentation can be after 2 years or maybe later. Need of the transfusion maybe never it is required, or sometimes it's occasionally required. So this diagram shows the chromosomal location and structural organization of the globin gene cluster. The upper part showing the beta globin gene cluster, which is present on the chromosome 11. You can see on this cluster, there is a beta gene, delta gene, and there is a pseudo beta gene also, which is non-functional, 2 gamma genes and epsilon gene. This epsilon gene making the embryonic hemoglobin, 2 gamma genes are making fetal hemoglobin, delta gene is making adult hemoglobin, and beta is also making adult hemoglobin. The same way, there is a promoter region which is known as the LCR regions. All these regions are important because mutations can occur in any part of this particular cluster, and finally they are going to play the role in the expression of these particular globin genes. Alpha globin gene cluster is present on the chromosome 16, where you can see there is 2 alpha genes are present, alpha 1 and alpha 2. They are almost 99%, they are similar, whereas 2 pseudo alpha genes are also present. They are non-functional. This is a structure of the beta globin gene. Beta globin gene is a 146 amino acid long. It's a very short gene where you can see that only 2 intronic regions are there, and exon 1, 2, and 3 are there. Basically, exon 1 and exon 2 are the hotspot region for all the mutations are located in these regions. Whereas exon 3 is basically a region where mostly dominant mutations occurs. And basically beta thalassemia is such kind of disease where all kind of mutations are there. Basically, it's a frame shift mutation, missense mutation, truncating mutations, splice site mutations. So the mutations in the intronic regions are also important because they occur at the splice site region. And the promoter regions mutations are also important because some of the mild mutations are also there. So till now, more than 350 mutations in the beta globin genes have been reported worldwide. And the type of mutations are beta 0 mutation, beta plus mutation, beta plus plus mutation. Beta 0 as itself showing by the name that there is a zero synthesis of the beta globin gene. Beta globin gene is not at all synthesized. Beta plus means there is a little deficit in the beta globin gene production, but it's not up to that extent that that is going to cause too much of effect. Beta plus plus mutation is a milder form of where deficit will be little lesser. These are the location of the different kind of beta globin gene and type of mutations occur on the clusters. Most of the nonsense frame shift mutations are beta 0 kind of mutation, and most of the mutation which are present at the promoter region of the genes and polydinylation tail, they are the beta plus sort of mutation. So beta gene mutations, the commonest in India, that definitely spectrum is differs from region to region and ethnicity wise, plus the population, whether they belongs to any consanguineous family or non-consanguineous one. But one thing one has to take into account that approximately 20 beta thalassemia mutation account for more than 80% of the beta thalassemia mutation across the globe. The difference is lying on almost rest of the 20% of the mutations. So when you can see that common mutations in India and globally also up to certain extent is IVS15, 619 base pair deletion, codon 4142, codon 89 and IVS11. Whereas less common mutation, they are codon 16, 15, codon 30 and codon 30 and codon cap plus 1 minus 88 and minus 25 deletion, which is present at the end of the IVS intervening sequence first. This particular map showing the differentiation of the mutation in the different regional areas of the India as per showing by the scatter diagram. You can see that blue part showing the IVS1 fund followed by yellow graph 619 base pair deletion. So most of the states are having top 5 mutation almost common, whereas less common mutations and rare mutations are being differed, and they can differ region wise. North India can have different kind of mutations from the south. Now the alpha thalassemia, as a defect in the alpha thalassemia is known as that alpha thalassemia, and the prevalence would be 10 to 18% in non-tribal population, because in tribal population this prevalence is way more high, whereas 10.5% are the single gene deletions reported and 2.3% two alpha gene deleted. Because in the alpha thalassemia, most of the mutations are not the point mutation, they are very less. Most of the mutations are the deletional kind of mutations. And in case alpha thalassemia is co-inheriting along with the beta thalassemia mutation, this can change the alpha beta globin ratio, and this change in the ratio can either make the phenotype severe or it can ameliorate the disease phenotype as per the number of alpha gene present in that particular patient. So in case because 2 alpha genes are present, so on both the chromosomes, so in case total 4 copies are present, this is the first diagram showing the normal genotype of the alpha. Second, showing the trait where 1 gene deletion is there. Third one and fourth one is there where 2 different gene deletions are there of the alpha. Whereas HBH disease having 3 alpha gene deleted and hydrops fetalis, which is the fatal form of the alpha thalassemia, there all the 4 genes will be deleted. And there is one kind of mutations which is known as alpha triplication, where none of the gene will be deleted, with, though one particular alpha gene will be increased because of the irregular crossing over. So there will be 5 alpha gene. The type of gene deletion in Indian population, the most common gene deletions are 3.7 kb deletion and 4.2 kb deletion, which are spanning both alpha 1 and alpha 2 genes. And here in this table, you can see in case the 4 alpha genes are there, the patient is absolutely healthy and 100% production of the alpha chain is there. In case 1 alpha gene is deleted, 75% alpha gene will be produced and these patients will be silent carrier. Whereas 2 alpha gene will be deleted, almost 50% of the alpha gene will be produced, and these patients you may see the change in the hematology, CBC and hypertrophic microcytic anemia will be observed. 3 alpha gene deleted HBH disease, 25% production will be there and may require blood transfusion in case major hemolytic anemia would be observed. Whether hydrops fetalis, where 0% alpha gene will be produced. This diagram showing the single gene deletion, you can see number of alpha gene deletion has been observed globally. They can span only a single alpha gene, whereas this diagram showing both the alpha gene deleted in number of mutations which have been observed globally. Now the genetic modifiers of the beta thalassemia. I would like to focus on the variable phenotype of beta thalassemia that is not only based on the beta thalassemia itself, but coexisting different type of set of diseases. Like first thing, type of beta thalassemia mutation. If the beta thalassemia mutation is beta 0 type, beta plus type, or beta plus plus type. Second, if number of alpha globin gene, what kind of alpha globin genes inherited is there. In case it is a deletional type of mutation or triplication, this can change the phenotype up to certain level. Co-inheritance of hemoglobin structural variant is also there, because most of the time the HBS, E, D Punjab, these structural variant, when they are co-inheriting with the beta mutation, generally patient represent as a thalassemia intermedia phenotype. And genetic factors that maintain high hemoglobin, fetal hemoglobin levels, they can reduce the severe phenotype up to certain level. So change of the phenotype in the beta thalassemia patient in the presence of the alpha thalassemia. In case the first lane, you can see if the patient is beta 0 or beta plus type of phenotype, and patient is not having any kind of alpha thalassemia, patient definitely will represent as a thalassemia major. Whereas the same patient, in case that is having the beta 0 type and beta plus plus type, but having two alpha gene deleted, there in this case both the alpha and beta chains are getting deleted, and they can compensate for each other, and the phenotype will be slightly milder, and patient can represent as a thalassemia intermedia. Whereas in the thalassemia trait, where only one gene is defected, be it beta plus type or beta 0 type, in case none of the alpha gene is deleted, it will be representing as a trait. And in case in the same patient, one alpha gene will be added or triplication is there, the same patient, due to the imbalance between the alpha beta chain, it will represent as a thalassemia intermedia. Now the HbF regulator, because many of the deletional type of thalassemias are there, delta beta thalassemias, HbFH, they are responsible for the maintaining high fetal hemoglobin level. Whereas non-deletional HbF regulators are linked to the beta cluster. There is a G gamma and A gamma promoter mutations. They are responsible for the high level of fetal hemoglobin. Whereas unlinked to beta globin cluster, there are certain loci which have been identified, and this is BCL11A or HBS1L, which are responsible for the high level of beta thalassemia. HPFH and delta beta thalassemia, most of the are the deletional mutation you can see here. Whereas examine 1G gamma polymorphism, this is one of the most important HbF regulator, which is being detected diagnostically also in the patients. This is in case this polymorphism is positive, this used to maintain and associated with the high level of fetal hemoglobin, and co-inheritance of examine 1 with the beta thalassemia can ameliorate the disease phenotype. These are the number of quantitative loci which are affiliated with the high level of HbF. Now, the structural hemoglobin variant, as I said, HbS, HbC, HbE, HbD Punjab, these are few of the common hemoglobin variant we identify in the lab day-to-day basis. And inheritance of these hemoglobin variant with the beta globin gene mutation may represent as a thalassemia intermedia. So now what is the need of the molecular diagnosis? First of all, because the first thing first, confirmatory diagnosis, because clinical picture and HPLC alone do not going to confirm the diagnosis of the disease and not reliably predict the mutation type. So for the confirmation diagnosis, you have to get it done, the molecular diagnosis. Plus, sometimes the HbA2 levels used to come in the indeterminate range. In that range, you can't just say the patient is thalassemia trait or something else. Again, the accurate diagnosis you will get from the molecular diagnosis only. The precise mutation detection, which type of mutation is there in the patient, you can only get from the molecular diagnosis, and then which can play a role in the prognosis of the disease, because you can get to know whether this is beta 0 mutation or beta plus type of mild mutations. The carrier and family screening, that can be done by the molecular methods, because the couple at risk for having the affected child, they can be counseled and they can take their decision of planning the family accordingly. Whereas carrier screening is also there, person will be aware about their carrier status and they can make the choice of their life partner, whether they are going to marry with the affected person or not. Prenatal diagnosis, again, one of the most important part in the thalassemia, where a molecular diagnosis can inform the couple at the early stage whether the bearing child is going to be affected or trait or absolutely normal, or they can take their decision accordingly to continue with the pregnancy. Pre-implantation genetic diagnosis. This is the diagnosis where during the IVF, when embryo has been created, at that time, that embryo can be checked for the mutations and chromosomal abnormalities by molecular methods, and then you can check and choose healthy embryos and can implant for the further pregnancy. So traditional molecular method, which is being utilized in India or everywhere, any lab which is engaged with the molecular diagnosis of the thalassemia must have been used any of these methods. These are all PCR based method. The most common one is allele specific PCR or ARMS PCR, which you can design for the known mutation and you can detect the mutations easily. Multiplex PCR, you can just more than one target can be detected in the same PCR. Reverse dot blot PCR is a very old method. Many targets can be run in a single go. Less time consuming also. Gap PCR generally being utilized in all the labs for the deletional mutation, especially for the alpha thalassemia. Whereas real-time PCR, you can design for the known target and you can see in the real time whether the amplification is taking place or not. All these methods are rapid, cost-effective, and basically meant for the known mutation, not for the unknown mutations or novel mutations. So the better approach for the, as compared to the conventional PCR would be the sequencing. The Sanger sequencing still remains the gold standard for identifying the globin gene mutations, because not only you are going to know the known mutations or common mutations which are prevalent in Indian or any other scenario, but unknown mutations also, rare mutations also, or any novel mutation also you can get which has not been reported till now. Everything you can get from the Sanger sequencing. So these are some of the representation pictures of the reverse dot blot, which is initially being done on the membrane, which is not no longer available. Now the reverse dot blot has come into the strip assays, the ARMS PCR and the DNA sequencing. The advanced approach, which are the upcoming things, and many people are using this thing also, is the next generation sequencing, because the technique is the high throughput and that can sequence many amplicons simultaneously so that you can detect common, rare, and novel mutation effectively. NGS can be two types. You can sequence the whole exome also, so that you can check all the things, all the not only beta mutations, but other genes also present in the genome. And the targeted sequencing, where you can go for all the genes involved in the beta thalassemia, beta gene, alpha gene, gamma gene, and including the tertiary modifiers like HFE gene, which is responsible for the iron overload. Anything you can cover in the targeted sequencing and make a panel and can run for the thalassemia. Though nowadays a third generation sequencing has also come, which is the long read sequencing, where the longer deletion of the alpha thalassemia, which was being missed by the conventional next generation sequencing, is being picked up by the third generation sequencing, because this is much more renowned and is a long read process. MLPA, again, this is a very nice method where you can detect a number of copy number variations like deletion and duplication in the globin gene, basically being used by the alpha thalassemia, because you can see I have put in two diagrams of the MLPA where you can see the deletion is shown by the red dots. That means there is a deletion in the alpha globin gene. You can easily detect the deletional mutation from the MLPA. Now, the prenatal diagnosis. Till now, many people were using the invasive procedures for the prenatal diagnosis, like chorionic villus sampling, amniocentesis, as cord blood sampling for the prenatal diagnosis. Whereas now it has been almost completely replaced by the non-invasive prenatal diagnosis method, which is known as the NIPT. This is a cell-free DNA isolation from maternal plasma, where you can get the cells of the fetus from maternal plasma. It's a non-invasive procedure. And because of this procedure, there are so many benefits. Like, it's a non-invasive, it's early detection you can do as early as the 9th or 10th week of gestation. High accuracy is there, reduced anxiety of the patient is associated, because, and this NIPT you will do for the chromosomal aberrations which are associated with the many of the thalassemic patients, like trisomy 21, trisomy 18, trisomy 13, and this can be done by the NGS. The pre-implantation genetic testing, where I have already told that the embryo which is being produced by the IVF, they can be tested previously, before even implanting to the uterus, they can be tested by the next generation sequencing so that they can identify whether any abnormality is there, be it related to the chromosomal abnormality or any thalassemic mutation or any other monogenic disease mutation. Then healthy embryo you can choose and implant. PGT is 3 types. PGTA for the aneuploidy detection, where chromosomal aberrations are associated. PGTM, this is for the monogenic disorders mutation. PGTSR, this is a new term which is for the structural rearrangement, where big deletion or translocations and duplication deletions are associated with the chromosomes. So the pre-implantation genetic diagnosis, the flow is like this. First of all, the IVF procedure is going to produce the embryo biopsy will be done, then PGTA or M will be done by the NGS, and then embryo selection will be done and implanting to the female's placenta. So at the end, I would like to tell what would be the clinical application and futuristic directions for the thalassemia diagnosis and management. Certainly accurate molecular diagnostic with the enhanced NGS or MLPA or digital PCR, which is also coming the way, because the cost is getting down and faster analysis is there, and their added advantage. The diagnosis should be prompt. Prenatal diagnosis, NIPT, and pre-implantation genetic diagnosis for the informed decision, so that individual can get to know what kind of the baby the lady is bearing, what kind of disease they are holding, or they can take their decision on the basis of the NIPT or PGT results. National screening program for the awareness and for the prevention of thalassemia, and this can also be done by the molecular screening. And the gene editing and gene therapy, as Dr. Swati told, this is altogether a different set of topic to be discussed, where gene editing and gene therapy is there for the management part. So the ultimate goal for the thalassemia is prevention of thalassemia, because it can't be cured, awareness of thalassemia, and improve the quality of life of the thalassemia patient. Thank you for the patient listening.
Thank you very much, Dr. Vandana, for the wonderful talk. I hope the participants were, got more insight about the genetic determinants and molecular diagnostic advancement in the field of thalassemia.